Hello, my name is Reza Talei, and today we are here for another SIT podcast. It's a great pleasure to introduce you to Dr. Iris Eke. Iris, please introduce yourself to the audience.
Yeah, so my name is Iris Eke, and I am working as a postdoc at the NCI in Dr. Coleman's group.
Okay, Iris, we would like to know more about your research. What are you doing there?
So the main focus of our group is radiation therapy-induced targets. So the idea is that you irradiate cells and then you examine the molecular changes which occur, and then you try to target these changes to kill tumor cells.
Okay, so you're looking for radiotherapy targets. Mm-hmm. How do you do your experiments? Do you look at cell line response, or do you do animal study?
Mm-hmm. So the first experiments we do in cell lines, and we use mainly solid tumors, and a lot of work is done in prostate cancer. And then after we have the results, we want to confirm these results in animal experiments.
Okay, so the animal experiments will come later.
Right.
Yeah. So you have an MD degree, and where did you get your MD, and then how did you become more interested from the clinical work to basic science?
Yeah, so I did my med school in Munich, and I was looking, because in Germany you don't get a doctor when you don't do an MD thesis. Okay. You don't get it just with your exam. So I was looking for an MD thesis, and one which I found was in radiation research, in which I was not so interested at this time, because it sounded very much like physics, and I was not so keen on physics. But when I did the MD thesis, I found it really very interesting. So because it's like a mix of technical things and also of biology. Yeah, and I think also because a lot of patients get irradiated, I think it's also very clinically relevant.
Yeah, and you had the opportunity to choose to practice MD or become a radiation oncologist or do research.
So the thing is, after my med school, I was so interested in research that I decided to do a PhD thesis first. So I went mainly into lab work, but after finishing this, I started residency, and I did this for 1 year and 3 months.
Okay.
And this was mainly in the hospital, giving chemo radiation to patients, and especially dealing with the side effects, which are sometimes very, very hard.
Mm-hmm. Where did you do that? Did you do it in US or in Germany?
In Dresden.
In Dresden.
Mm-hmm.
Okay.
Yeah.
Okay, and that was after your PhD?
Right.
And then you decided to move to US.
Yes.
How did you find your project?
So the thing was, my significant other moved to the NIH. Okay. So he's also a doctor, a physician, and he got a position at NIH. So I was looking mainly in the DC area.
Okay.
And because my former boss, Michael Baumann, had contacts to Dr. Coleman. Okay. So he asked, and Dr. Coleman was happy to take me.
Okay. And you're continuing on your postdoc, but are you considering to go back to clinic, do a residency in US, or are you considering to follow your research route?
Yeah, so at the moment I'm doing a postdoc, but my aim would be to do a residency, because I would really like to combine the research with clinical work and go more into clinical studies, and therefore I want to do residency. It's not so easy for a foreign person, not only because you have to do the exams again, but also radiation oncology is of course very competitive here, and as a foreign student it's sometimes a bit more difficult.
Yeah, so let's go back to your research. Mm-hmm. What has been prominent in your research? What did you like more? What publication you have done that you like? Can you explain a little bit more about the science you do?
Mm-hmm. So in my PhD thesis, what we did was we developed 3D cell culture model. So the idea is that because when you do in vitro experiments, it's often that you can't transfer these experiments to in vivo studies, but you can't do everything in vivo because it's just too much. So it would be good to have a more advanced cell culture model. And we developed a 3D cell culture model where you can also do clonogenic survival, which is very important for radiation research.
Yep.
So I spent a lot doing this and also validating these data. And because the 3D model, the integrins are very important. So the main topic was the role of integrins for the radiation resistance of different tumor cells.
Nice. And in your postdoc, you're targeting some molecules. Can you explain about them? What are their role and how do you target them?
Yeah, so I also use a 3D cell culture model at NCI because we have seen that when you target cells and you use in vitro 2D or 3D, the cells react very different, and often the 3D model better reflects the in vivo situation. So the idea was that we look in 3D at the molecular changes of cells after radiotherapy, and for this we use mainly fractionated radiotherapy because at the moment, it's changing now, but at the moment most patients are radiated with a fractionated regime. And then we found that after fractionated radiation, prostate cancer cells got, the AKT-mTOR pathway got activated after fractionated radiation. And then we checked when we target these molecules after radiotherapy, is it more effective than when you target it before radiotherapy? Yes. And we found that it is a cell line dependent effect, but if you target them when the pathway is activated, the drugs work very much better.
Okay, nice. And what did you bring here to radiation research to present?
So this project,
This was the project that you brought.
Yeah. So I think it's also quite different because most or a lot of people examine the effects of targeted therapy as a radiosensitizer, so therefore you give it before. And the idea of our project is so maybe you can sensitize cells with radiotherapy against the targeted therapies. So it's the other way around.
And are you involved in other projects that you want to explain about them to us?
Yeah. So I have two main projects. So one is a targeted therapy project, and the other is the idea is that when you radiate single cells with a single dose or with fractionated radiotherapy, normally cells survive better when you give fractionated radiotherapy. Okay. So maybe they can repair between and survive better. And the idea is that maybe the DNA repair protein or the DNA repair processes are different after fractionated radiation compared to single dose, because you have a lot of changes in the chromatin, and it could be that the DNA repair is different.
So is this work focused on tumor cells or is it on normal tissue?
So the first idea was that we use knockout cells, murine knockout cells, and compare if there is a difference between single dose and fractionated radiation. And then we used these experiments or these results and transferred these to cancer cells. So we did a knockdown in cancer cells and looked if these results were also there.
So before I come to my final question, if you think there is anything interesting in your research, both in the scientific part or in the other part, like collaborating with people, or anything that you want to explain, please go ahead and then share the idea with us.
So the thing is, at NIH you have very nice collaboration possibilities. So for example, the DNA repair project wouldn't be possible without collaboration partners because of the knockout cells, for example, and other reagents. And I think, so radiation research, it's very important that you always have, like, at least for the medical part, it's always important that you have a connection between medicine and biology. And I think especially in the Radiation Research Society, I think this is really very, very good. Because I think when you want to do translational research, you have to have both.
Nice. And my final question is that where do you see the future of your research going towards?
So I would like to do more clinical studies with the results I have I have now from my lab. It would be, of course, the last aim is to improve cancer therapy really, really in patients. And my goal would be to try if this concept of target induction is also possible in clinical studies.
Nice.
But maybe that's a long way.
Yeah. Thank you very much for your time, and we enjoyed talking to you.
Yeah, thank you very much for the interview.